All right, we're going to get started in a couple minutes. Just, I know people are trickling in and grabbing lunch, but we want to stay on time. So I have the tremendous pleasure today to introduce two people. One is Dr. Mark Nesbit. This is the 24th annual Mark Nesbit Lectureship. And for those of you who don't know Dr. Nesbit, he is sitting right here. He spent his career here and really built the Pediatric Oncology program, and very importantly, built what has now become the Children's Oncology Group, starting from working together and realizing that to make advances in pediatric cancer, we needed to work together and collaboratively, and started with the Children's Cancer Group, and that then became merged with the Pediatric Oncology Group to become the Children's Oncology Group. Dr. Nesbit also was the first North American president of PSYOP, the international organization that works together and collaboratively to study cancers in children, and really paved the way for international collaboration and work within pediatric cancer. And also importantly, Dr. Nesbit lent his mark on 3 diseases that changed the course of history. He ran the first national trial for Ewing sarcoma and really paved the way for proving that chemotherapy makes a difference in that disease. He led the studies that demonstrated that intrathecal chemotherapy for children with leukemia actually improves outcome, and without his studies, that would not be part of our therapy today. And very importantly, he paved the way for studies in a rare disease called histiocytosis, and really was the pioneer in the treatment of that disease. So left his mark in many, many ways. And we today are very blessed to have him with us and to continue to learn and to continue to benefit from his wisdom, which we did this morning with lots of great discussions with Dr. Malkin and questions with Mark. So this year, as the 24th annual lectureship, I am pleased to have with us Dr. David Malkin. He's a professor at the University of Toronto Hospital for Sick Children in both pediatrics and genetics. He runs their cancer genetics program there and has developed really a world-leading expertise in genetic predisposition syndromes for childhood cancers, and particularly an expert in the Li-Fraumeni syndrome. And so he is here today to share with us many wonderful, I think, years of work and overview, and how collaborative work has really paved the way in these rare diseases. So thank you, David.
Thanks very much, Brenda. And I've got lots of microphones here, so I'm not sure which one I'm going to use. I think I'll just use this one.
Yep, you can that one. Yeah.
All right. You know, with the piano and everything, I feel one of these days some grand round speaker is going to sit down and play for you. It's not going to be me. Not me. No. It's been a long time. So I'd like to thank Brenda and University, the hospital, for inviting me. This is an absolutely wonderful honor. And Dr. Nesbit, we had breakfast today and really enjoyed sort of going back over time and looking forward. And this really is an honor for me to be here and speak at this lectureship. And it's been also a pleasure to meet some old friends, Dr. McMillan, Dr. Ramsey, and many others, and as well as new people. And the session that we just had on some cases leading to what we hope will be a very nice manuscript coming out of one of our fellows here, Rachel, wherever you are. I'm going to keep pushing that. And but that's what this is all about. So it's been a wonderful 2 days. I've really enjoyed it, and the weather has helped, of course. So I'm going to spend the next 40 minutes or so giving you a somewhat whirlwind tour of Li-Fraumeni syndrome. And I think there are probably some of the residents here who are saying, what is that? And others who are saying, so how can you spend the next 45 minutes talking about it? But this is, as Dr. Wiles mentioned, a paradigm example of the value of studying and understanding rare diseases in children or in humans, and applying that with our ability to integrate interesting research questions and bring it to a full circle where the research is basically defining the clinical disease and the management of it, and the clinical observation is defining the research questions. So I don't have any disclosures because I'm a pediatrician and we just don't have financial disclosures. But last Friday, I'll just give you an idea of what I do in my clinic. So I actually have a cancer genetics clinic, which I combine with a solid tumor clinic. And last Friday, I had a family with familial hereditary paraganglioma pheochromocytoma syndrome and have a mutation in the 6-enol dehydrogenase genes, one of the genes. All 3 children in the family are carriers, and we're following them. 2 kids with von Hippel-Lindau disease. One had developed a pheochromocytoma and the other a renal cell carcinoma. And a child with a cystic nephroma, and we were just talking about this with Dicer 1 syndrome, so a relatively new syndrome, only defined about 5 years ago, but exploding on the pediatric oncology stage, as you will. And another kid who had a pleuropulmonary blastoma. Again, rare stuff, you think, except it's telling us an awful lot about human biology and cancer biology. We saw 3 families with Li-Fraumeni syndrome, classic families where the children are unaffected, but we're following them with surveillance, which we'll talk about a little bit. And then another 2 kids who are carriers of the mutation, one with metastatic adrenocortical carcinoma who is finishing her therapy currently, and one with a rhabdomyosarcoma who's actually just completed therapy, and we're following. So it's a real mixture of activity that goes on in a very interesting and exciting program. But it leads then to this syndrome, the so-called Li-Fraumeni LFS Pedigree. And the 2 individuals at the top there, on your left is Fred Lee, and on the right is Joe Fraumeni. And they described it in 1969, so it's going back quite a ways. And they described it based on a review of patient records of about 600 children with rhabdomyosarcoma, looking for family history, and identified 5 of them that had a mother with breast cancer and then other cancers in the history. And they published this at that time, but it wasn't really understood what might be causing it, and there was no such thing as DNA sequencing, so there was really nothing to look for. But over the next 20 years, they followed about 24 families that led, as you'll see in the next slide, to a definition. This is one of those typical families, and I know that you probably really can't see the little writing under each of the squares and circles, but basically what it is to highlight is that you get multiple malignancies. We'll see in a couple of slides the spectrum of cancers, brain tumors, adrenal, lung. I mean, they don't seem to have anything in common. And the ages of onset are typically very young. So usually in the adults, they may be 20 years younger than what might normally see for sporadic tumors. And in children, they might be usually in their first decade of life. So very early onset, multiple cancers. You have to think about Li-Fraumeni syndrome. Before I go any further, I just want to highlight Fred, and I hope I get through the next 30 seconds okay. Fred passed away 10 days ago. He was a mentor to me. I mean, there are many mentors that we have in our careers, and this is really directed at the residents and fellows. We all should have many mentors, but there are always some mentors who somehow or other are, if you will, almost iconic. They're the individuals that you think of as perhaps pursuing not only your career, as Fred did. He had a brilliant career in science and medicine. He'd been ill for a number of years, so his death, while sad, was not unexpected. But more important than that, he really taught me and all of those who trained with him how to live and how to live life, and to be able to balance one's career and one's life and one's family life and one's friendships. And this is probably, I think, what most of us who knew him well will remember him most for. But it is a sad point in our, at least for me, because we were very, very close. I could pick up the phone any time of day or night, ask him questions, and know that I would get a incredibly depth response. So a scientist, a physician par excellence, a mentor, and most importantly, a lifelong friend. So just thought I would put that out there. Li-Fraumeni, challenges. Taking on Fred's legacy, what do we have to do? Well, all of those cancers that we have no idea how to prevent them from developing. We don't have any idea how to prevent multiple cancers from developing. These patients get multiple tumors typically over their lifetime, sometimes 5, 6, 7 malignancies. And we think that therapy has something to do with that, maybe accelerating it. We talked a little bit this morning earlier. We don't really know how to predict the types of cancers nor the ages of onset in which they'll develop. And we also really don't have effective treatments beyond what we would typically use for any of these malignancies. But it's even more challenging than that because they develop multiple tumors, you often are using up a lot of your best therapies early on. And there's also a challenge that you might actually be causing tumors by the actual use of the therapies we do. So for the next half hour or so, 40 minutes, this will kind of be the outline that we have. We'll talk a little, bring everybody up to speed on the syndrome and on the gene that's associated with it. We'll jump right into this surveillance protocol, the idea of being able to at least impact the outcome of patients if possible. And then we'll delve a little bit more into genetics. So we'll start a bit clinical and then dive into some of the molecular stuff and then see where we end up at the end. So the syndrome, as I mentioned, was described in 1969, but it wasn't really defined until about 20 years later. And this is the definition that is generally considered classic. You need a sarcoma somewhere in the family in a young person, multiple first and second degree relatives. And the point at the bottom comes up shortly is that about 50% of individuals will develop a second tumor, assuming that they survive their first one. So the risk is lifelong of developing multiple malignancies. The tumor spectrum is quite wide. In fact, almost every cancer has been defined in a Li-Fraumeni syndrome somewhere. And until about 2.5 hours ago or so, I would say that the two tumors that really had not been described are Ewing sarcoma and hepatocellular carcinoma. Now we can reduce that to Ewing sarcoma. So thank you, guys. But this is exciting. And this actually goes to the point that a young child with a now hepatocellular carcinoma, we will likely want to change their practice and consider P53 mutation testing. It's never been done, and it's the classic example. If you don't look for it, you're not going to find it. And to be fair, I don't think any of us were looking for it in HCC after probably not finding it in a few patients. So nonetheless, it's going to be rare. So what are the common tumors? Well, the common ones are about 25% of individuals, women, will go on to develop breast cancer, generally in their 20s and 30s. Another 25% of individuals will develop sarcomas, both bone and various soft tissue sarcomas. About 20% develop brain tumors. Virtually every type of brain tumor has been described. And then there's a smattering of other early onset tumors. But attention should be drawn down to the bottom corner here, where I've listed so-called other component tumors. And what I mean there are, if you look at that list, there's lung cancer, ovarian, colorectal, melanoma, a whole bunch of adult cancers. Well, in fact, these cancers don't occur in kids in Li-Fraumeni syndrome, but they occur about 20 to 30 years younger than they would typically be expected in a sporadic, as a sporadic tumor. So if you see a child with a rhabdomyosarcoma at age 3, whose grandfather perhaps, maybe the parents may be okay, but the grandfather died or developed a lung cancer at age 40, then you should at least think that this could be a Li-Fraumeni family. Because lung cancer shouldn't happen at age 40, particularly if it's a non-smoker. So these are the kinds of questions that we follow and we ask and expand the story. Now, as I mentioned, it's a pretty highly penetrant disease, and in fact, about 3/4 of males will develop cancer in their lifetime, and almost 100% of females. And that extra risk is primarily the breast cancer in females. So it is the most highly penetrant genetic disease we know. When you take the women in particular, in cancer at least. Now the other thing is, as I mentioned, that we have a very high risk of second malignancies, an 83-fold increase in developing second malignancies. And this will likely increase as treatment for primary cancer in children particularly improves. As the more of those patients survive, they are going to go on and develop second, third, fourth malignancies. So this is really quite a devastating disease. And what's also interesting about it is that you can look now, there's large enough numbers of families. There's several hundred, probably several thousand reported in the world's literature, and they're really now being able to dive back in. So people who aren't interested in the molecular stuff might be interested in epidemiology, and there's a very rich population of patients to work with. So a molecular epidemiologist named Pierre Reynaud, who's in France, in Grenoble, France, he's been mining the various data sets that are available, and it sort of came up with this diagram of 3 phases of the disease. So there's a childhood phase, which we obviously are very interested in this group, where there's a very high incidence of cancer compared to the general population in the green from the SEER database up until about age 20. And about 30 to 40% of people with the mutation in this gene will develop cancer before they're 40. It's very much in our bailiwick of activity. Then there's a second group of adults, young adults, with another peak here. Mainly this red one is the women with breast cancer, but there's another sort of phase there. And then there's this older phase of individuals where, interestingly enough, the incidence of new cancers drops to below the general population average, suggesting almost that if you survive long enough, there may be a protective effect of carrying this mutant gene, which we really don't understand, but the data would suggest that something unusual is happening. It's less frequent than even in normals. Now, the only main problem I have with this slide, which you probably pointed out, and I'm sure it was lost in translation, is apparently if you're over 45, you're considered to be elderly. I take great, anyway. So, who is this? Well, if you saw the title of the talk, this is Damocles, all right? And the idea of why I put this slide up here, which I stole actually from Josh Schiffman in Utah, is the story of Damocles, where he basically had asked, he said, to the king, it's an easy job to be a king. You can do anything you want. You can order anything you want. You order your people around. It's an easy job, right? And the king said, well, okay, so you be king for a day. And he realized that there was the Damocles sword, that there was always something sitting over you, that there was a potential disaster ready to happen. And that's what being king or queen or President Obama is all about. There's always something around the corner that you've got to be ready for that you don't know. And this is what Li-Fraumeni syndrome is about, because these people live day to day wondering if in them there's a cancer growing. And they don't know about it, and they can't do anything about it. And so we think of this a little bit like Damocles' sword. All right, so Li-Fraumeni. Now we'll switch over to P53. Very interesting gene. This is the protein structure. It's a molecule that has forms 4 monomers, a quaternary structure, binds to the double helix of DNA, and activates downstream targets that are involved in various processes. So it's a classic tumor suppressor most of the time. It's obviously, well, not obviously, but it's 53 kilodaltons in weight, which is where the number comes from. And as I mentioned, it binds to DNA. And it activates a whole slew of cell regulation activities, including DNA repair, apoptosis, everything that's sort of listed there, plus many other things. Metabolism, too, has now become very a hot topic. P53 in tumors, in cancers themselves, are mutated in about 80% of human cancers. In pediatric cancers, the ones that we see in a place like Children's here, that is probably more like 10 to 15%. It's not that commonly altered in most childhood cancers, but it's very commonly mutated in the adult world. But the pathway that P53 regulates is inactivated in a wide variety of malignancies. So even though P53 itself might be intact in pediatric cancers, often the pathway is involved. Now, mutations in the gene are what basically inactivate or disrupt that ability to bind DNA. And as may not be a big surprise, as I showed you here, the thing is bound to the double helix there, so it's probably not a big surprise that most of the mutations actually do occur in the DNA binding domain. And so you can imagine if you have a mutation at this little green amino acid 282, interestingly enough, that was one of the amino acids we talked about today, then it disrupts the ability of binding, and so you're not able to activate all these good genes that are transcribed down the road. So about 6 or 7 hotspot mutations that account for about 40% of all the mutations in the gene. And what happens is that when that mutation occurs, then all of the potential things out there that can induce DNA damage or genomic instability, you've lost the guardian. P53 is the guardian of the genome. It protects the genome from dividing with damaged DNA. And so if you've lost that ability, then you can imagine DNA in the cells continue to divide, and those are obviously setting up for malignant transformation. So all of things that induce DNA damage are protected with intact P53 to activate all these other processes, but if P53 is inactivated, then all of this is lost. So it's an incredibly important gene in normal human development. Now, going back for a second then, we had made this discovery back in 1990 that germline P53 mutations account for a significant number, about 80% of these classic families I showed you in that definition with Li-Fraumeni syndrome. But over the years, we've seen many families that don't quite fit the definition as it was described, and so new criteria were developed by the French group, and Champré was the original author of the first paper. And so they've been kind of given her nomiker. But these are sort of the definitions we have. And this is actually a typo. That should be 46 years of age. But basically, there's some similarities here. And I just point you down to this last row. A child who presents with an adrenocortical carcinoma or a choroid plexus carcinoma, rare tumors, albeit, should be considered for testing for P53 regardless of their family history, because at least 50% of them carry a germline mutation. And this has implications not only for the child, but of course for the entire family. So these are the sort of new current criteria. We'll see as we go on that we're going to be changing them some more. And then this is the spectrum of mutations that occur in the germline, similar to where they occur in tumors themselves. Most of them clustered in this area, which is the DNA binding domain I showed you. And then there's one really oddball mutation out here, which is in the tetramerization domain of the molecule, which allows it to form tetramers. And that mutation, codon 337, has only been described in Brazil, and actually only in the southeast portion of the country. It's thought to be a founder mutation, and there's an amazing story about some guy who came from Portugal back in the 1600s, and he was a merchant, and he landed in the northern part of the southern provinces, and he was a merchant. So he would go down, and he'd stop in different villages along the way, and he would sell his products, and he would also deposit other things. And so gradually over the centuries, there are these spectacular families with this mutation that have been carried through, and they're all related to this tropeiro, who is my lousy Portuguese for a merchant. And he definitely caused problems. I should say that the carrier rate was about 1 in 2 to 5,000 in the general population. It's about 1 in 300 in Brazil. So it's a very, very common problem. All right, if you want a short version, easy to read, on the, when you're coming into work or sitting up late at night, you don't want to read medicine, you can read this book by Sue Armstrong, who's a freelance writer, and she wrote a book about P53, the gene that cracked the cancer code, which I have no idea what that title's supposed to mean, but anyway, she interviewed a whole bunch of people who were involved in the field, and read it at your pleasure. You can find it on Amazon.com for $20. It's not exactly on the bookshelves or your recent stores, but it's there. It's a kind of a fun read of the story. Most of it's true. Most of the personalities are, well, I won't say anything about them. Okay, so we'll have breakdown into 4 or 5 stories. The first is, can we do anything with these families to perhaps change the course of their disease? And so about 5 years ago, a resident at the time, she's a pediatric resident now, just completing her fellowship, in oncology, Anita Villani, worked with me and our team on developing and then implementing this protocol to see if we could come up with a way to identify tumors early. And so it's a surveillance protocol for individuals, children, and the next slide, adults, with germline mutations in P53. And we're looking for the tumors that are most common in the disease. So adrenocortical tumors, brain tumors, soft tissue tumors, leukemia, lymphoma, and just kind of generally everything else. And we've developed this protocol using a variety of biochemical and imaging studies to see what would happen. Similarly, in adults, where we added the breast cancer screening as well as colorectal cancer screening, because those tumors occur a little bit later than the pediatric age group. And with that, and this is taken really from the paper, was published a few years ago, 33 patients were basically, they weren't randomized. You can't randomize in a study like this. They basically self-selected to be surveilled or not. And about half of them turned out to have no surveillance, and half of them turned out to have surveillance. It should say that most of this group were the children, and almost no children were in the no surveillance group, because parents want their kids to get surveillance. But you can see here that all the patients at the end of that particular follow-up period were alive, and at that time was about 72 months of follow-up. Well, you could say, well, maybe they just didn't develop tumors. But it turns out, in fact, that they did at an almost equal rate. So the number of tumors presenting in asymptomatic patients who were being screened was about the same as those in the symptomatic patients who did not get screening and just presented to their doctor with something. And so when you break it down that way, we see that, in fact, the survival rate obviously is similar. And the tumors themselves are not all benign. They're actually malignant. All of them are malignant tumors. And in fact, most of them occurred in children, which suggests, in fact, that this is the population that we may be able to have the most impact on, because the vast majority of them in that initial group had, for therapy, relatively little therapy. Almost all of them were able to be put into long-term remissions and potential cures of their primary tumor with surgery alone. One had 4 cycles of low-dose chemotherapy before surgical resection of a choroid plexus tumor, and one had myelodysplastic syndrome. She ended up going to transplant because she had MDS, and the discussion was just to take her to transplant straight up. But one might argue that she at least went to transplant before transforming. So we don't know that for sure. Since then, there have been a number of other patients followed and more tumors developed, and there's now this protocol is used in various places, including here. And at the time we published it, this was the data. And for an oncologist to be able to get Kaplan-Meier curves where the survival is 100% in anything is just never seen. And in fact, it isn't seen, because in the longer-term follow-up, we were putting this together. One of those patients has actually died of metastatic disease. So we know that it's not perfect, but we do hope that it has improved. And we'll explain towards the end of why we think it might actually have an impact. So now we'll talk, so this is actually one of the patients. This is a patient in Detroit who had an asymptomatic lesion picked up on screening, a small rhabdomyosarcoma in the maxilla. She was referred to our center. One of our surgeons is really a world-renowned for nerve grafts and such, and so they worked on her to resect this so that she would avoid radiation, which we were able to, I shouldn't say we, the royal we, he was able to do. And this is 2 days after after surgery, and you can see that she is grimacing, and the reason she's grimacing is because I gave her a Toronto Blue Jays hat. But then a few weeks later, she was playing golf, and she was having a fun day at Lake Michigan on the beach, and she had some chemotherapy, but at least it was Vadriac, vincristine, actino, and cyclophosphamide, most of which are felt to be fairly safe in the context of this gene. So we now know the protocol is used all over the world, and in deference to being here in Minnesota, I've stuck a big X, and I realize it's not exactly Minnesota there. That's probably more like Kansas, but, you know, I'm from Canada, so please excuse me. All right, so the conclusions are that the clinical surveillance protocol does detect pre-symptomatic tumors, and perhaps in conjunction is improving survival, long-term survival, and we shall see if this progresses and how it goes along. But it is still not personalized. It's still not personalized. Everybody gets basically the same protocol. So I'm going to give you 4 scenarios that are each clinical tidbits to try to understand a little bit more of how we can use these things. So as I mentioned before, this is a CT scan of a patient who presented to us with an adrenocortical carcinoma, and it's awfully similar to the one that we just saw in rounds or in the case presentations, which is a huge, huge adrenocortical carcinoma, okay? And this was a patient actually presented with metastatic disease at age 9 months, all right? So for those who are less familiar with it, it's a bad disease if it is caught when it is late. So if you have a small adrenal tumor, the overall survival is outstanding, but even with toxic chemotherapy and years worth of chemotherapy, really anybody with poor stage disease does really quite poorly. So Jonathan Wasserman, who's an endocrinologist at our center, but had been doing a postdoc with me in the lab until he was recruited on board, was interested in this disease, and together with the Children's Oncology group, there's a clinical trial that we were involved with, wanted to explore this gene and this disease more in depth. So the first step was to look through the IARC. This is the International Agency Against Cancer based in France, and they have a very large database of P53 stuff that they've collected over the last 20 so years. And from that was able to extract about 160 individuals who had germline mutations in P53 and characterize them on a various demographic parts. And what was interesting, at least one of the observations, is the frequency of tumors in these mutations tended to be very high early on, and in fact, almost 70% of individuals who developed adrenocortical carcinoma did so within the first 4 years of life, very young, and in fact 90% do so by their 20th birthday. So in the context of a germline mutation, this is a very pediatric disease. And you'd say, okay, well, that's very interesting and that's fine, but in fact, when you compare it against what typically occurs in a sporadic adrenocortical carcinoma, that's a disease of older people. Elderly people, if you will. Okay, so it is definitely a disease of older people, whereas in the context of a germline mutation, it's a disease of very young people. It's a different disease, and it gets a little bit, I think, to Dr. Nesbit's question to me before, is can you sort of decide what is going on, or can you differentiate different diseases by knowing something about the P53 status? And this is a clue that we probably can. So what Jonathan did is, with the COG registry, as well as our own patients that we collected over a few years, looked at 89 patients under the age of 18. We had a number who had this Brazilian mutation, and we excluded them from the analysis. So about half of the patients with germline mutations, half of the patients with adrenocortical carcinoma as children had a germline mutation. That itself wasn't novel because that had been reported by the French group. But what we did find was interesting is only one of those patients, the mutation was in that hotspot I described. So there are all weird mutations happening across the gene that really, some had been described, but some were novel and some didn't repeat themselves. In fact, only 3 were recurrent in unrelated individuals. So each patient seems to have a little bit of a different story, which of course makes life a little bit complicated. They were all young, and we'll have one little slide a little bit later, but basically other polymorphisms within P53 or involved in P53 function were entirely non-correlated with the presence of the mutations. So what he did is went along and did some functional work looking at cells that have deficient P53 to see if there was any way to sort of distinguish the role of the mutation with the phenotype. And basically, for those who don't know, these are colony-forming assays where this would be a cell line that has no P53, so the cells are growing like crazy, and there you see them all there. And then if you stick wild-type P53 back in, you basically kill them all, or they stop dividing. But then you can use different mutations and find out that, in fact, you have various degrees of growth, so that might be of some value. But the other thing which was particularly interesting is he looked at the activation, transcription of all these downstream targets that I mentioned before, P53's function. How is it activating and working on other genes? And if the wild type is here at 100% P53 activity, you can see there's a group that have almost no activity, there's a group that sort of have reasonably good activity, and there's a group that actually are like super repressors. They have better activity than P53. And these are all the different mutations along the line. And what we was able to show is actually almost all the mutations that occurred in patients with aggressive tumors, a strong family history, multiple malignancies. These were all ones where P53 was completely wiped out. It just was not working at all. Whereas patients who basically had isolated tumors and not much else had, if not functional P53, at least very mildly abrogated P53, suggesting that the type of mutation may actually tell you something about the type of cancer or the phenotype that is going to go along. So what this means for this little story is about half of patients with this tumor have germline mutations. The mutations have a wide spectrum of activity, and whether they're sufficient to cause disease, we don't really know, but you'll see in the next story that they probably are not, okay? So this is the next story. Medulloblastoma, another tumor that is frequently seen in these families. So this is a young boy, 14 years old, who presented originally with a tumor back here in the cerebellum that was a medulloblastoma that our neuro-oncologists had anticipated having a very good survival based on various parameters of the tumor. But it turned out that he recurred rapidly and died of his disease, and his family history suggested a Li-Fraumeni family. His mom had had breast cancer, there was a sarcoma over here, and a variety of other cancers as well. And so he had a P53 sequence and had a mutation in the gene. All right, so shifting slightly across the pond, otherwise known as the Atlantic Ocean, there was a group in 2011 at Cambridge University that was doing whole genome sequencing of viable tumors to see what sequences were like, and this is something that, of course, is being done all the time. And they found in a small sort of fraction of cancers generally, there was this process, what they call chromotripsis going on, which is basically a single catastrophic event where you induce a recombination of two chromosomes, sometimes three, with end-to-end rejoining of the ends. And it happens, it's thought to happen basically at once. So it's not an accumulation of events, it's one event happening at one particular time. And they found it in a sort of a subset of tumors. So this is sort of the difference. Normally we think of cancer coming from multiple genetic alterations, rearrangements, mutations, and eventually the genome gets totally screwed up. Whereas in a catastrophic model, it's one event and you end up with shattering of chromosomes, and some of them come together in some non-homologous kind of end-joining process. So why was this of interest? Well, Jan Korbel, who's a scientist in Heidelberg, was sequencing medulloblastomas. He knew nothing about Li-Fraumeni syndrome. He was interested in brain tumors, pediatric brain tumors, so he was sequencing several dozens of these medulloblastomas. And what he found, to his surprise, that in a subset of them, they also showed this chromotripsis. We're talking in the tumor itself, not in the blood, in the tumor. And so he said, well, that's kind of interesting. And it turned out that all of the patients who had chromotripsis in the tumor also had a mutation of P53 in the tumor. And so there was a little bit of a question, could the mutant P53 somehow or other have something to do with this weird catastrophic event? So, and on the flip side, none of the ones with no chromotripsis had a mutation. So it's pretty clean a differentiation between those populations. So what we had known about this work, and he had known about us, and so what we did is collaborated, because we had collected blood and tumor. He only had tumor. We always collect blood and tumor on every single one of our patients, not because we know what to do with it, but because we know that at some point someone's going to ask a really interesting question, and at least we'll have the material. So we collected them, and we happen to have a reasonable number of medulloblastoma patients, and all of them, when you looked in the germline, had a germline P53 mutation. So what it suggested was a very early mutation in this gene had something to do with this catastrophic event. And that was kind of interesting, and we'll see right at the end, the second last slide, why we think it's important for surveillance. The other thing, however, is that all of those medulloblastomas were of this, what we call sonic hedgehog type. There's 4 types of medulloblastomas based on their molecular profile, and one of them, so-called sonic hedgehog, that interesting developmental pathway, they were all the ones in the same group, so that when we looked here, this is sort of the 4 groups of medulloblastoma. This is a sonic hedgehog, and you see all the red dots. Those are all the Li-Fraumeni patients. They're all clustered together. So you need, appears, that if you have a germline mutation and you don't have in the tumor a sonic hedgehog mutation, you won't get a medulloblastoma. But if you do have a sonic hedgehog mutation in a brain cell that's going to be potentially programmed to medulloblastoma, you will get this, and you'll get it because you have chromotripsis. And this is just for those who look at sequences all the time. This is chromotryptic sequencing. It just doesn't look normal, even if you've never seen one of these before. It's just a completely shattered kind of region of the chromosome. So this little story tells us then that germline mutations confer a susceptibility to chromotripsis, probably not sufficient, and at least in the context of medulloblastoma, sonic hedgehog mutations. The third story is another tumor we see commonly, which is rhabdomyosarcoma. Okay, so this is a young boy who presented at 19 months of age. No, he's a little less, sorry, 14 months of age with an orbital rhabdomyosarcoma, which you can see here. His mother had just completed therapy for a phylloides tumor of the breast, so breast carcinoma, and he came in and has a strong family history of cancer. He was tested, as was mom, and they carry a mutation in P53. There are two main subtypes of rhabdomyosarcoma in children, embryonal subtype and alveolar, and I'm not going to belabor this for the moment, but there's another subtype too called anaplastic rhabdomyosarcoma, and it's called anaplastic because the cells themselves look anaplastic. They look much more disrupted and dysregulated, and they actually tend to behave a little bit poorly. Now, this had been known for some time, and what's interesting is if you stain an anaplastic rhabdomyosarcoma in the lab with P53, it lights up like a Christmas tree, or at least a very brown Christmas tree. And so what that indicates is P53 is stabilized in the cell, and mutant P53 is stable. Wild-type P53 is not. So you know that the P53 is mutant. So just keep that in the head for a second. Well, we had previously published back in, I don't know, mid-90s that children under the age of 3 with rhabdomyosarcoma had a likelihood, about 10% of them, 15%, had germline mutations in the gene, irrespective of family history. And over the years, others have also shown similar things. So a group in Boston, including Lisa Diller, who's one of the oncologists there, had actually worked with us on that project. And in about the spring of 2014, she had called me and said, you know what, we just got 2 patients within about a month of each other with rhabdomyosarcoma who had germline P53 mutations because they were young. But both of them had anaplastic variant. And I said, boy, that is weird. So what we did is we went into that database again, that bank again, which we had collected for some reason, but now we had a reason. So we went back into our database of rhabdomyosarcoma samples and pulled out all the ones that had germline P53 mutations done because they were young, and that was the reason they were done, and all of them had anaplasia. Then we flipped the story around and we reconsented a bunch of patients who had had their rhabdomyosarcomas banked but had not had germline testing for whatever reason. And we reconsented them to see if they would consent to testing, or their parents who consented them to testing. And when we did that, it turns out that 75% of this set had germline mutations in P53. So all of a sudden it shifts that Champré criteria because now a child with anaplastic rhabdomyosarcoma, like the little guy that I just showed you, has a germline mutation. Why is that important? Because one of the questions for this kid in terms of therapy was, do we radiate that eye, or do we exenterate the eye? Surgery or radiation? Ideally radiation, because you preserve the eye. But in the context of a germline mutation in P53, we felt, after lengthy discussion with the family, that a surgical intervention was probably safer in the long run, and that's exactly what he had. And you can see that here. While he was getting his chemotherapy, vincristine, actinomycin, cyclophosphamide, we continued surveillance because he still had Li-Fraumeni syndrome. And he presented asymptomatically on his second MRI scan with an adrenal lesion there. Turned out to be an adrenocortical carcinoma. His blood work also was slightly elevated at the time. We gave 1 course of chemotherapy for that because the surgeon felt he would be able to shave it off the IVC would be tough, and he subsequently had it resected, and he's done very well since then, and he has no other problems, except that not that long ago on another surveillance scan, we found that he has what has been biopsy proven to be an orbital low-grade osteosarcoma. That one we're keeping an eye on because it's surgically extremely difficult to deal with. But he's well, he's running around, and he is a maniac on wheels, and he's 3 years old. So, rhabdo with anaplasia is now part of the syndrome. Germline mutations might contribute to these karyotypes. We don't know anything about this. I just put that up there. And that we also don't see alveolar rhabdomyosarcoma very frequently, so there might be some protective event going on. And then the last disease that we see commonly in children is acute lymphoblastic leukemia, right? Most common childhood malignancy. So this is borrowed from Charles Mulligan at St. Jude, who published a huge amount on the genome, the ALL genome, and this just sort of all the various molecular events that occur in childhood ALL. And I just point you down to this one here. About 1% of them are so-called hypodiploid ALLs, and these tend to do quite poorly unless transplanted. So they also tend to occur at an early age. They tend to be much earlier in this little group here, under the age of about 4 or 5. So they tend not to be leukemias of the older or adolescent and young adult. As I mentioned, they're relatively infrequent, but they have this interesting characteristic of the hypodiploidy, which is important because what they did is the St. Jude group, screening a large number of ALLs generally, found that 85% of the hypodiploid leukemic blasts, so not the germline, looking at the blast, had a mutation in P53. It was actually the most common mutation seen of all of the others in the near diploid or near haploid. Well, that was kind of interesting because then what it turned out, the mutations are spread all over the gene, in fact, is that almost half of them had that mutation in their germline. Now, the germline was measured either from remission marrow. I had suggested to them that they perhaps look for another tissue because it could be possible that they just have a minimal clone. So they also did fibroblasts as well as buccal swabs, and there was consistent. So we know that this is real. So what it means then is that you can see this syndrome continues to evolve. And just over the last 2 years, we've added 3 totally unexpected diseases. And in addition, now virtually every woman with early onset breast cancer, when they are undergoing genetic testing, places either do a panel or they do individual screening of the BRCA1 and 2 genes, but also P53 is now included almost always, and a portion of them, somewhere around 8 to 10%, will have a germline mutation. Okay, in the last 7 minutes or so, I'm going to outline some of where the biology is going. So what this slide does is summarize a very large amount of work done by a lot of different groups, trying to address the question is, are there modifying genetic events that could help us in kind of creating an algorithm that we might predict what types of tumors or what ages of onset occur in individual patients within families. And to this point, we haven't really got to the algorithm yet, but we have a number of particular targets. So some of these things like this duplication and this polymorphism are within P53. This gene is involved in P53 degradation. And then there are these sort of more genomic variations as well as microRNAs which are involved in P53 regulation. And you can see that all of these things that have been described in a variety of families and studies have something to do with age of onset, but not a whole awful lot to do with the type of cancer that they're developing. So we might be getting closer to be able to predict when an individual will develop cancer, but we're still not really very much further along of what type that might be. So everything here now is unpublished data, and what one of the MD PhD students in our lab, Nardine Samuel, has been pursuing for the last 18 months or so, almost 2 years, is to look at the methylome. And the question is it possible that there are epigenetic events occurring in the environment of the cell or in the environment locally of the cell or of the individual that are impacting that cellular's ability to respond to the presence of a mutant P53, which may presumably have biological effects? So using the database that we have, she took about 92 mutation carriers and about 100 age and so on matched controls, and after a variety of deletions and exclusions, came out with about 70 individuals that she could analyze with about 100 controls, and then a validation cohort of about half of them. And what we're doing here is looking at the blood of these patients, all right, looking at peripheral blood lymphocytes, and she's also matched it against some skin fibroblasts. Spectrum of tumors is similar to everything else we see. A little bit of a predominance of adrenocortical tumors is because we seem to collect them, but everything else is much as we would find. And there's also a group of patients who carry a mutation but have not yet developed cancer. So there are various ages in that regard. But it's an interesting group because we wonder, why is it that some people never develop cancer? And a good question. So using this 2x2 square, she's been doing a variety of analyses. I won't go through all of them by any means, but the one that I want to spend a moment on is, sorry, that's just kind of the fancy stuff that she uses, which I'm not going to bore you with because, frankly, I don't understand some of it, but just to show that it is done. And what she did is two analyses. One is to take those patients who had partially functional P53 in their germline. So remember that adrenocortical histogram. There were these individuals who had mutations that really didn't disrupt P53 function very much. So that's that group against those that had wild-type P53. And this volcano plot basically shows these is areas of demethylation of the genome and degrees of hypermethylation of the genome. And basically there is no significant difference between the two in this plot. However, when she looked at deleterious mutation carriers versus wild type, there is definitely a number, there's actually about 49 sites across the genome that are differentially methylated between those that had deleterious mutations and those that are wild type. And there's one up there, which is called MIR34A, microRNA 34A, which I'll talk about for a moment, because it is actually significant to about 10 to the minus 17 or so. So MIR34A, it's a microRNA, very interesting area of work, of course, in many fields. But this was first characterized a few years ago as the first one that is transactivated by P53. And its inhibition leads to a variety of functional dysregulation of P53, which are important in cell cycle and apoptosis, the key main functions that this gene have. We've known that it's hypermethylated in many cancers. Nobody's ever looked at the methylome in the germline, but its real mechanism of how this works is still pretty poorly understood. So Nardine has gone on and validated all of this using first of all, this is just the validation cohort to see that this is actually real, and also in individual families. This particular family is exciting in the sense that we have actually material on everybody, and those with a black dot are mutation carriers, those without are not mutation carriers, and you can see that those with the mutation have generally a hypomethylated germline, if you will, compared to those who don't. Very, very distinct, suggesting there is a direct correlation of some sort or association. This heat map is a little bit difficult for those who aren't totally familiar with it. Here we've listed all 49 genes that appear to be differentially methylated, at least significantly, down this axis, and here are all the various samples across the top. And what you can see in the box here, you can see there are patterns of differential methylation between this group and that group. You can just looking at the color difference, you can distinguish that. And what's interesting is in this particular group, those, all with the black dots, are patients with P53 mutations. And there are far more in this grouping than there are here, again, correlating with this. But what's even more potentially exciting is that, again, we have all the tumors placed across the top. Here we have a larger number of genes down the side. But here we actually have the tumors. And so in pink, for example, are patients who have brain tumors, whereas over here in green are patients with adrenocortical carcinoma. And the patients who are developing tumors have a different methylation pattern in their germline, which seems to be somewhat predictive of at least the type of tumors that they eventually develop down the road. This has some real potential, we think, as a predictive marker for the type of cancer, which then leads to the type of surveillance one might do. So the summary at the moment, this is obviously work in progress. We have a manuscript that's being developed, a lot of functional work going on, but it does suggest that there may be epigenetic alterations which actually work together with P53 to define the tissues that are most targeted depending on where the demethylated site or hypermethylated site might be. And so we're doing a variety of studies to pursue that. So in the last minute or so then, we'll just summarize. So there was a model that we had proposed a few years ago looking at the concept that if you have a mutation in the germline, you have some copy number variation of your genome, which in the germline, sort of you more rapidly develop genomic instability and so more rapidly develop mutations in various cells that might transform. And what we now think might be happening is that you are either born with or acquire a number of variants in the germline, which interact with P53 in one way or another. And these interactions probably render, if not the genome more unstable, they definitely render the defense mechanism unstable, more inefficient for P53 to work. And some of that may be reflected in differential methylation of binding partners, in something I didn't talk about at all, accelerated telomere attrition, and of course in chromotripsis, which is basically, if your genome is so unstable, it's going to shatter all over the place, right? As long as P53 is not there to protect it. And when that happens, then you basically get mutation-specific, cell-specific mutations causing the cancer. And the idea might be that maybe if we have the surveillance coming in here, you're able to actually stop the process before those malignant changes, those dramatic changes occur. So the tumors that are picked up, at least they are benign or low grade, and you're able to reverse biology. So we have a few answers, but lots of questions. Didn't talk at all about this, but we talked a little earlier about ethical and psychosocial implications of testing. We have no idea about the economic impact. There's a study going on to determine is surveillance better than just treating. We don't know what to do with the therapy question, and being fairly blunt, and we don't really know what the biological mechanisms, though we think we're getting closer into what causes these things. So at the end of the day, those 4 challenges I mentioned at the beginning, we're closer, but we sure haven't addressed them or completely answered them. Okay, so we reached the fork in the road. I'm sorry, I'm happy. I know Margie was going to laugh when I say. All right, so the fork in the road is this. This is this little girl I mentioned from Michigan, and last September, she entered her first day of school in her hometown. And in December, her mom gave birth to a little boy who was tested, and he has a wild type P53, so he's not at risk. But unfortunately, mom had breast cancer, and she relapsed. And so this is the ultimate challenge we're facing, are these families, and we talked about a couple of them today. This is probably the most devastating kind of disease that I have ever run across, because it never goes away at all, and it has just got its tentacles everywhere. And so this is what we're faced with. And to address it, we put together a large team in Canada of investigators, both at our place and in Halifax, to really come up with new ways perhaps that we can address the problem. So we have mathematics and sequencing, and seeing there zebrafish, and imaging. And what we're doing is combining expertise from a variety of disciplines. So there's me working at the lab, yeah, right. Then Andrea Doria, who is an amazing radiologist, is developing novel techniques using MRI imaging to more rapidly and more sensitively identify lesions in patients. Adam Schlein is a genomics guru who is doing some amazing modeling of the genome of tumors, as well as some of the germline stuff we're doing. And then together with Anna Goldenberg is developing this multi-level approach to be able to develop these predictive algorithms that might be ultimately useful. But at the end of the day, wouldn't it be amazing if we could actually prevent these cancers from developing in the first place? To do that, we've engaged a really exciting guy, Jason Berman, who's in Halifax, where they've got lots of fish, even though it's cod, but in his lab, they've got zebrafish. And so he's modeling Li-Fraumeni fish, and will be exploring exposure to various chemicals. And so by doing that, we have this multidisciplinary team, which is now funded in its totality by the Terry Fox Research Institute, which some of you may be familiar with. He's the guy 35 years ago who ran halfway across Canada to raise money for cancer, and has now raised about close to $1 million. Sorry, that's the team. Now, if you understood absolutely nothing about what I told you, then I would like to refer you to this particular episode of Grey's Anatomy. So, anybody here actually saw this? Oh, good, Logan. Thank you. All right. Okay. It's interesting. None of the residents and fellows watch this. That's where you get all your medical knowledge these days. Come on. I mean, we used to do that with ER. Yeah, ER was the way I did, or St. Elsewhere. But anyway, so on this particular episode, the entire story is around Li-Fraumeni syndrome. They covered everything, and of course, the surgeons did the genetic counseling, genetic testing. They got sequences in a few hours, so they figured out how to treat them. But at the end of the day, it was the intern, I don't remember which one, who came up with an interesting way to treat the patient. And none of us in the Li-Fraumeni field claim any responsibility for any of the content of that episode, just in case you were wondering. that is it. This is my team at, I guess, just this past Christmas. And this is actually the much larger team, and many folks outside the institution as well, many of our funders, and most importantly, are an incredible group of families who are incredibly altruistic and giving of their time, of their blood, of their tumors, and I thank them most especially, and I thank you.
Thank you, David, for a great talk. That was really fun. And it's fun to see the evolution and to how all of these incredible, like the last slide, that huge international collaboration of trying to put together this work is phenomenal and a tremendous tour de force. So thank you. I think we may have a couple minutes for questions if anyone has any burning questions.
That was great. You didn't address this at all, but I saw in one of your slides it looked like a family had PGD. Are you getting that, and how is that addressed? And
right. So we do get families requesting, if not PGD, at least prenatal diagnosis as an option, and a number have utilized that to either elect a therapeutic termination of the pregnancy, or at least know at the time of birth. PGD, we've had a few. The problem for us is of all the wonderful things in the Canadian healthcare system, PGD is actually not covered. Everything else seems to be, but not PGD. So there have been a couple of families that have actually, and insurance, or the private insurance generally don't cover that as well up north. So they have gone elsewhere and had it done. Interestingly enough, in France, it's covered entirely. PGD is covered entirely for all sorts of genetic diseases. We're not quite there yet, but we hope so.
Cost-benefit analysis
of
any disease.
This is one. This is one. But there have been other scenarios where the moms have said, or the dads have said, they've gone right through the pregnancy, they lived, and they said, if my mother had made that decision, I wouldn't be here today. So it's that kind of ethical balance that we play with. Yeah.
Great talk. I was really fascinated by your methylation data because I've always thought of Li-Fraumeni working through loss of heterozygosity, right? And here you're showing some level of cellular dysfunction in unaffected carriers. So do you see other cellular dysfunctions? And I'm wondering why there isn't a Li-Fraumeni phenotype, that you see with other syndromes, if indeed it's working in haploinsufficiency somehow.
Yeah, so it's a very important point because, in fact, it has been traditionally thought that there's a haplosufficient genotype. But in fact, about 40% of the mutations actually are not gain-of-function mutations. So there is actually retention of the wild-type allele, and it's not a complete loss of function allele, so that it's not truly a haplosufficient scenario. In terms of the biology and the function, we actually are working on that now, so we don't have a whole awful lot of data. But I should say that I thought, and I just realized I meant to put a slide in, where one of the other students is actually looking at the tumors, not so much the germline, but the tumors. And what she's shown is that there is actually a completely different pattern of methylation using the 450K between individuals who have, in the tumors, between individuals who have a germline mutation and those who do not, suggesting that there are actually secondary events occurring in the tumors that may program, if you will, the pattern of methylation in the tumor itself. And this has been in adrenocortical carcinomas as well as rhabdos, which is the largest subsets we have, as well as choroplexus carcinoma. So we think that the event that might be occurring in the germline is actually carried through to the tumor. What we haven't done is sort of dissect what particular sites are up and down, yeah, but hoping to get there.
So I was really curious to see the information on hypodiploid ALL. And as you know, they're really poor actors usually, and they usually
You really had to ask that question, didn't you? So probably the most common question, clinical question we get is exactly that. What do we do in the context of TBI for transplant, or actually radiation even outside, focal radiation for a tumor? But to get to the TBI question, I think what's happened, there's not a great deal of data in the literature one way or the other to suggest that TBI is good or, well, I shouldn't say good, but bad or worse in these patients. But theoretically, and as well as in mouse models, there's absolutely no question that if you do equivalent of TBI in a mouse with the P53 mutation, they accelerate tumor formation. So, and we've seen in a couple of patients with MDS who gone on to transplant, they've had accelerated disease. We've seen it with CCNU and a variety of other chemo agents. What we've generally done as a group is suggest that you have to ultimately treat the disease sitting in front of you with the best possible state-of-the-art therapy. If there is an alternative to TBI, then absolutely. But often the alternatives are activated through P53. So you're probably going to end up in the same boat. It's just it's chemotherapy instead of radiation. So I know it's a bit of a hand-waving argument, but we generally have said if it is the only way to do it, then you kind of got to do it. Otherwise, you're going to lose the child for their hypodiploid ALL. But as I mentioned to the group before, one of the patients that I'm seeing that we're doing a neoplastic tumor board on tomorrow, referred from Ottawa, had hypodiploid ALL at the age of 2. This was before Mulligan's work. And then 10 years later, had an AML, was transplanted from the brother. At that point, they had tested, and the brother was wild type, but the kid has a mutation because they decided to test. And then a year later now is presented and now has an osteosarcoma. But the question was, she had TBI at the original transplant, but that's 10 years. I'm not quite sure if those are connected or whether the second AML is just truly a second tumor. The reason I think it's the latter is because she actually has loss, getting to Logan's point, she actually has loss of the wild type allele of P53 in the AML, suggesting that the second hit, the AML may have been primarily caused by the P53, not so much by whatever she was treated up front. But you wonder about the osteo. Yeah.
Hi. I have another question along those lines of radiation and treatment. Is there much research available with regard to the CyberKnife radiation treatment and the proton beam radiation?
So again, not a whole awful lot of zero literature and not a lot of experience other than occasional anecdotal reports of people using it and running into trouble or people using it and not running it into trouble, but they're isolated cases. We don't believe, and I don't believe really there's all that much difference between proton, cyber, or IMRT. In this day and age, they're able to, of course, narrow the field as they would normally do, and all of them pick off normal tissue. So there's two questions there. One is it more likely to induce a second malignancy? Absolutely. I mean, protons cause the same, the mechanism's the same. So if there is normal tissue picked up, then at least that is possible. And we don't have enough data to know whether the actual scatter radiation dose makes a difference in the context of P53. There is some data in mice, but how well it extrapolates, we don't know. The second, though, and very important question is that the tumors themselves are P53 mutant by definition. They have no, unless they retained a wild type allele, there is no wild type function. So now you're actually radiating a lesion which is not able to repair the DNA that you're destroying with or damaging with the radiation. So unless it's a lethal dose of radiation to that cell, the likelihood of it recurring in the context of that radiation is actually extremely high. Just in all principles of biology would say that. And we have definitely seen patients who were radiated. They didn't develop a second tumor in the outside the field. They came back with a vengeance. In the recurrence comes back with a vengeance. And I think it's because, of course, the lethal dose is not universal around that tumor. There is going to be our cells which are either slightly protected or receive a sublethal dose, and those ones are not able to repair. They just take off. So that's another challenge with radiation is unless it's lethal, you probably are setting the cell up for this is a real problem. Yeah, real problem.
So thank you. I think we've had great questions. If there are more, I'm sure you can come on up, but I think we have hit our time limit on the room. But I would like to thank, again, Dr. David Malkin for coming and visiting us from the Hospital for Sick Children in the University of Toronto. And again, to thank Mark for the legacy of now the 24th annual. Next year will be the big 25th, so stay tuned. Mark Nesbit Lectureship. And thanks again, David, for a great lectureship and a great visit.